Immunotherapies have a unique potential to improve the treatment for cancers while not significantly increasing toxicities. The key to this is the immune system's ability to very accurately define what types of cells it's seeing and therefore what should be attacked and what should be left alone. The T cells are a very active part of the immune system, and they are responsible for identifying tumor cells and killing them. Too often, however, our immune systems don't recognize cancer cells, and so our work is in an attempt to help the T cells learn to distinguish tumor cells from normal cells. In order to improve immunotherapies, however, it's going to be necessary to combine them with other therapies that really increase the inflammatory immune response. The danger of that is if the targeting of the T cells is not well done, we could increase toxicity significantly. So this work was an attempt to find tumor-specific proteins that are created by tumor cells and not by any normal cells that can be used to help target those T cells so that their toxic abilities are trained completely on the tumor cell. It's our hope that this specificity is going to improve the efficiency and efficacy of the T cell killing without increasing toxicity, and therefore creating a platform in which combinatorial immunotherapies will be more possible. Our strong hope that the ability to distinguish these tumor cells so specifically will make it possible for us to train a patient's own immune cells to attack their tumor. We're moving quickly towards a clinical trial in which a patient's tumor will be removed and characterized to identify these tumor-specific proteins. And then their same immune cells will be taken out and trained to recognize those proteins. After that, the T cells will be reinfused into the patient so that they can go and seek out tumor cells and kill them. We're also working on a methodology to improve the creation of these T cells that target the tumor-specific proteins. This approach is not dissimilar from an mRNA vaccine such as has been made for COVID, except in this case, we do it in a dish rather than in a patient. We take dendritic cells, which are also part of the patient's immune system, and we put the mRNA to create these tumor-specific proteins into the dendritic cell. Then the dendritic cell makes those proteins itself, processes them, and presents them to the T cells. The T cells who recognize that particular target will expand themselves by making copies. And in that way, we generate a large number of T cells that can create the therapy that we provide to the patient.